Our method is significant as it allows the average biochemist to quickly determine if samples isolated using immobilized metal affinity chromatography are contaminated with transition metals. The main advantage is the ease by which the assay can be performed. The assay uses instrumentation and techniques common to most biochemistry labs, and it can be easily and quickly implemented. While in this article we apply this method for samples isolated with a nickel NTA column, the method can be used with any other metal affinity resin. First-time users should try this method with a nickel stock solution of known concentration. This will familiarize them with the workflow, sample colors, and corresponding spectral changes. Demonstrating the procedure will be called Swaim, a technician from my laboratory.
To begin, turn on and warm up the UV-Vis spectrophotometer. Determine the chromatography fractions to be assayed using a diode array UV-Vis spectrophotometer with optical absorbance at 280 nanometers to quantify the protein. Obtain 10 to 100 millimolar sample buffer with a pH between 7 and 12, such as tris, heaps, mops, and phosphate buffer. Prepare a 12% weight by volume solution of HNB dispersion in the sample buffer using 120 milligrams of HNB reagent for each milliliter of stock solution prepared. Set the spectrophotometer to collect data at 647 nanometers. Use a quartz cuvette filled with the sample buffer to blank the spectrophotometer. Prepare a control solution in a cuvette containing 50 microliters of HNB stock per milliliter of